Hello friend. In this module, we are going to see different types of clinical trial designs. Though statistics is not directly related to the clinical trial designs, there is an indirect relationship between the designing the clinical studies as well as the statistics. So we are going to see what is the relation and what are the types of clinical trial designs one by one. Why during a clinical trial a design is important? In clinical research, our aim is to design a study which would be able to derive a valid and meaningful scientific conclusions using appropriate statistical methods. Before choosing a study design, one must establish aims and objectives of the study and choose an appropriate target population that is most representative of the population being studied. The conclusions derived from a research study can either improve healthcare or result in inadvertent harm to the patient. Hence, the study design requires a well-designed clinical research study that rests on a strong foundation of a detailed methodology and is governed by ethical principles. Now, we are going to see the different types of trial designs. First, about the blinding. There is unblinded studies or open label studies where the participants and the clinicians and all study stakeholders knows which treatment goes to whom, which patient. And then the single blind and single mask study where the investigator or the patient will be blinded and they don't know which treatment goes to whom. In case of double blind study, both investigator as well as the patients will not come to know which treatment goes to which patient. And finally, the triple blind study where the investigator and patients as well as the data analyst who is doing the analysis also don't know which data belongs to which treatment. So these are the types of blinding which is generally used for the clinical studies. And for the bioequivalent studies, generally unblinded or open label designs are followed. And in case of clinical trials or the clinical endpoints, where the double blind studies, where the investigator as well as the patients are masked and they don't know which treatment goes to which patient. Now it is the design configuration. So the design configuration there will be three design configuration. One is the parallel group design, where the treatments are assigned to different patients, where for example, treatment A assigned to subject A or patient A and treatment B assigned to subject B or patient B, like that. So there will be a different groups. One group will be assigned to treatment A and another group will be assigned to treatment B, another group assigned to treatment C. That is called parallel group design. And crossover designs where one subject or one patient will be treated for both treatments or all the treatments. So in a crossover manner, that is called crossover designs. Apart from that, there are factorial designs where there will be multiple administration of the particular treatments to the same subject at a different point of times. There are trial designs based on the type of comparison. So some trials which will show the superiority. So superiority over the placebo. For example, you are going to use one of the treatment is the drug treatment, another one is the placebo treatment. Then you have to show the superiority of the drug treatment against the placebo. Then there are some trials which show equivalence or non-inferiority, where you are establishing equivalence between treatment A and treatment B. Both are drug treatment. Then there are trials to show dose response relationship. And also there are group sequential designs where cross-sectional designs and longitudinal research designs are adopted. Now we are going to see the different trial designs with much more details. So now about open label studies. So what is open label studies? Clinical trial in which the subject, participant and the clinicians or the data collectors are aware of the treatment the participant receives. For example, bioequivalent studies. Then it is single blind clinical trial where clinical trial in which the subject or the participant is unaware of the treatment they are receiving. For example, a marketing person prepares several cups of cola labeled A and B. One set of cups has Pepsi, the other have Coca-Cola. A marketing person knows which soda is in which cup, but he is not supposed to reveal this information to the subjects. Now, double-blind clinical trial. In this clinical trial, neither the subject nor the observer knows which treatment is being administered. Double-blind methods can be applied to any experimental situation where there is the possibility that the results will be affected by conscious or unconscious bias on the part of the experimenter. Example, clinical studies that are conducted to test new drugs. In these studies, random assignment is used to allocate patients into two groups, the treatment or the experimental group and the control group. Participants and researchers are kept unaware of which participants are allocated to the treatment group. Now, triple blind clinical trial. In this clinical trial, the subject, the observer or the researcher and the researcher who analyze the data all do not know which treatment is being received by which patient. This is done when the clinical variables examined can be interpreted in different ways. For example, a triple-blinded, randomized, placebo-controlled trial to examine the efficacy of buspirone added to typical antipsychotic drugs in patients with chronic schizophrenia. The purpose of these studies was to test the hypothesis that the addition of buspirone, a partial agonist of 5-HT1A receptor, to ongoing treatment with typical antipsychotics would improve the positive and negative symptoms in patients with chronic schizophrenia. This slide talks about parallel group designs. The most common clinical trial design for confirmatory trials is the parallel group design, in which subjects are randomized to one of two or more arms, each arm being allocated to a different treatment. These treatments will include the investigational product at one or more doses and one or more control treatments such as placebo or active comparator. The assumptions underlying these designs are less complex than other designs. However, as with other designs, there may be additional features of the trial that complicate the analysis and interpretation. For example, covariates, repeated measurements over time, etc. There is an example. A two-arm parallel assignment involves two groups of participants. One group receives drug A, another group receives drug B. So during the trial, participants in one group receive a drug A in parallel to the participants to the other group who receive drug B. This slide talks about crossover design. In a crossover design, each subject is randomized to a sequence of two or more treatments and hence acts as his own control for treatment comparison. This design reduces the number of subjects and usually the number of assessments needed to achieve a specific power, sometimes to a marked extent. In a simplest 2x2 crossover design, each subject receives each two treatments in a randomized order in two successive treatment periods, often separated by a washout period. The most common extension of this entails comparing two treatments in number two periods, each subject receiving all two treatments. Numerous variations exist, such as designs in which each subject receives a subset of two treatments or ones in which treatment are repeated within the subject. In continuation to the crossover designs have a number of problems that can invalidate their results. The chief difficulty concerns carryover, that is the residual influence of treatments in subsequent treatment periods. For example, in the first period you are taking one drug and the effect of the drug or the level of the drug can be carried over to the second period during which the another treatment is being given. In an additive model, the effect of unequal carryover will be to bias direct treatment comparisons. In the 2x2 design, the carryover effect cannot be statistically distinguished from the interaction between treatment and the period, and the test for either of these effects lacks power because the corresponding contrast is between subjects. This problem is less acute in higher order designs but cannot be entirely dismissed. In this slide, we are seeing the difference between the parallel and crossover design. In a parallel study, you can see there are two groups. In one group which is given placebo and another group which is given the drug based on a randomization manner. In a crossover study, here you can see the period 1 and the period 2 distinguished by a washout period. And in the first period, the subject will receive the drug treatment, and after the crossover, you will have the second period, he will receive the placebo. Similarly, the subject who receives the placebo in the first period will receive the drug in the second period. So this is called a typical crossover design. This is also being done based on the randomized manner. In this slide, we are going to see factorial designs. In a factorial design, two or more treatments are evaluated simultaneously through the use of varying combinations of the treatments. In a simplest example is the 2 into 2 factorial design, in which subjects are randomly allocated to one of the four possible combination of two treatments, that is A and B. So what are the possible combinations? In one combination, we have A alone, in another one, B alone, in another third one, both A and B will be given, in the fourth combination where neither A nor B will be given. So in many cases, this design is used for the specific purpose of examining the interaction of A and B. The statistical test of interaction may lack power to detect an interaction if the sample size was calculated based on the test for main effects. This consideration is important when this design is used for examining the joint effects of A and B in particular, if the treatments are likely to be used together. Another important use of the factorial design is to establish the dose response characteristics of simultaneous use of treatments. For example, for treatment C and D, especially when the efficacy of each monotherapy has been established at some dose in prior trials, a number m of doses of C is selected, usually including a zero dose, that is placebo, and a similar number n of doses of D. The full design then consists of m into n treatment groups, each receiving a different combination of doses of C and D. The resulting estimate of the response surface may then be used to help to identify an appropriate combination of doses of C and D for clinical use. This particular slide schematically explains the factorial design, where here you can see the four combinations what we previously seen. In one combination we are seeing the X is given, in another one Y is given, another one both are given, and the last one nothing is given. So this is one of the factorial design. Another one is the combination of different treatments, where in one you can see the A plus C is given, another one is B plus D is given, another combination C plus D is given, another combination A plus. This is how the factorial designs are being used for the different studies. Now we are going to see about multi-center trials. Multi-center trials are carried out for two main reasons. Firstly, a multi-center trial is an accepted way of evaluating a new medication more efficiently. Under some circumstances, it may represent the only practical means of acquiring sufficient subject to satisfy the trial objective within a reasonable time frame. Means you have a huge sample size for which you need many sites, many centers to do the trial. And they may have several centers with a large number of subjects per center, or in the case of rare disease, they may have a large number of centers with a very few subjects per center. In continuation to multi-center trials, a trial may be designed as a multi-center trial primarily to provide a better basis for the subsequent generalization of its findings. This arises from the possibility of recruiting the subjects from a wider population and of administering the medication in a broader range of clinical settings, thus representing an experimental situation that is more typical of future use. In this case, the involvement of a number of investigators also gives the potential for a wider range of clinical judgment concerning the value of the medication. Such a trial would be a confirmatory trial in the later phases of the drug development and would be likely to involve large number of investigators and centers. It might sometimes be conducted in a number of different countries in order to facilitate generalizability even further. In the simplest multi-center trial, each investigator will be responsible for the subjects recruited at one hospital, so that center is identified uniquely by either investigator or hospital. In many trials, however, the situation is more complex. One investigator may recruit subjects from several hospitals. One investigator may represent a team of clinicians, that is sub-investigators, who all recruit subjects from their own clinics at one hospital or at several associated hospitals. In case of multi-center trials, the statistical model to be adopted for the estimation and testing of treatment effects should be described in the protocol. The main treatment effect may be investigated first using a model which allows for the center differences but does not include a term for treatment by center interaction. If the treatment effect is homogeneous across centers, the routine inclusion of interaction terms in the model reduces the efficiency of the test for the main effects. In the presence of true heterogeneity of treatment effects, the interpretation of the main treatment effect is controversial. This particular diagram typically talks about how a multicenter trial is being organized. So these are the different centers which is adopted in this particular trial. Center 1, Center 2 and Center 3, like that. Number of centers can be included. Each center have a different set of participants, and each center have the ethics committee, individual ethics committee, and each center will have a principal investigator. And entire setup is being coordinated by the particular CRO, and there will be a sponsor, and there will be clinical trial management group, clinical trial associates, there will be a data manager and analyst. There is a possibility that there will be a funding agency, there is a drug safety members group, and this entire setup is being completely organized by a multi-center trials. In this particular slide, we are going to see about the superiority trial. Scientifically, efficacy is most convincingly established by demonstrating superiority to placebo in a placebo-controlled trial, or by showing superiority to an active control treatment, or by demonstrating a dose-response relationship. This type of trial is referred to as a superiority. For serious illness, when a therapeutic treatment which has been shown to be efficacious by superiority trial exists, a placebo-controlled trial may be considered unethical. In that case, a scientifically sound use of an active treatment as a control should be considered. The appropriateness of placebo control versus active control should be considered on a trial by trial basis. So for example, a study that tests whether a new medicine for asthma works better than one that some patient already used. Or you can say in a study where the drug is cytotoxic and it is being used for a cancer patient, and in which a placebo cannot be used. This slide talks about equivalence or non-inferiority trial. In some cases, an investigational product is compared to a reference treatment without the objective of showing superiority. This type of trial is divided into two major categories according to its objective. One is equivalence trial and the other one is non-inferiority trial. Bioequivalence trials falls into the former category, that is equivalence trial. In some situations, clinical equivalence trials are also undertaken for the regulatory reasons, such as demonstrating the clinical equivalence of a generic product to the marketed product when the compound is not absorbed and therefore not present in the bloodstream. Many active control trials are designed to show that the efficacy of an investigational product is no worse than that of the active comparator, and hence fall into the later category, that is non-inferiority trial. Another possibility is a trial in which multiple doses of the investigational drug are compared with the recommended dose or multiple doses of standard drug. The purpose of this design is simultaneously to show a dose-response relationship for the investigational product and to compare the investigational product with the active control. Active control equivalence or non-inferiority trials may also incorporate a placebo, thus pursuing multiple goals in one trial. For example, they may establish superiority to placebo and hence validate the trial design and simultaneously evaluate the degree of similarity of the efficacy and safety of the active comparator. The equivalence or non-inferiority trial is not conservative in nature, so that many flaws in the design or conduct of the trial will tend to bias the results towards a conclusion of equivalence. Concluding equivalence or non-inferiority based on observing a non-significant test results of the null hypothesis that there is no difference between the investigational product and the active comparator is inappropriate. Now we are going to see about dose-response relationship trial. Dose-response trials may serve a number of objectives, amongst which the following are of particular importance. That is, that the confirmation of the efficacy, the investigation of the shape and location of the dose-response curve, then the estimation of an appropriate starting dose, then the identification of optimal strategies for individual dose adjustments, then the determination of a maximal dose beyond which additional benefit would be unlikely to occur. These objectives should be addressed using the data collected at a number of doses under investigation, including a placebo, that is zero dose, wherever appropriate. The hypothesis tests that are used may need to be tailored to the natural ordering of doses or to particular questions regarding the shape of the dose response curve. That is called monotonicity. This slide presents a typical dose-response relationship trial. This trial is a randomized double-blind dose-response control crossover study to evaluate the safety and efficacy of two doses of a drug A in chronic pancreatitis patients. The objective of the study is to evaluate the difference in coefficient of fat absorption of participants treated with high-dose drug A versus low dose of drug A in the treatment of signs and symptoms of malabsorption in chronic pancreatitis patients. Now we are going to see group sequential design. Group sequential designs are used to facilitate the conduct of interim analysis. It is most commonly applied because it is more practicable to assess grouped subject outcomes at periodic intervals during the trial than on a continuous basis as data from each subject become available. The statistical method should be fully specified in advance of the availability of the information on the treatment outcomes and subject treatment assignments. An independent data monitoring committee may be used to review or to conduct an interim analysis of data arising from a group sequential design. While the design has been most widely and successfully used in large long-term trials of mortality or major non-fatal endpoints, its use is growing in other circumstances. In particular, it is recognized that safety must be monitored in all the trials, and therefore the need for formal procedures to cover early stopping for safety reasons should always be considered. There are two types of group sequential designs. They are cross-sectional designs and longitudinal research designs. Cross-sectional research designs are used to examine behavior in patients of different ages who are tested at the same point of time. The researchers might choose to give a certain intelligence test to individuals who are 20-year-old, individuals who are 50-year-old, or individuals who are 80 years old at the same time and compare the data from each group. Longitudinal research designs. These designs are used to examine behavior in same individuals over time. For example, study of intelligence and aging. A researcher might conduct a longitudinal study to examine whether 20-year-olds becomes less intelligent with age over time. To this end, a researcher might give an intelligence test to individuals when they are 20-year-old, again when they are 50-year-old, and then again when they are 80 years old. This study is longitudinal in nature because the researcher plans to study the same individuals as they age. With this, we have completed the different types of clinical trial designs and the relationship between the clinical trial designs and the statistics. Thank you.